I'm David Smith. I'm from the University of Oxford. I am studying for my PhD in the Barnes group, in the Peter Medwar Pathogen Research Group. But what I'm here to talk about today is the work I did before that with Jim on using electronic patient records to investigate viral hepatitis. So first thing I just introduce you to viral hepatitis. So hepatitis is basically just an inflammation of the liver, and viral hepatitis is hepatitis caused by a viral infection. It can be caused by one of these five viruses, which are helpfully labeled A, B, C, D, and E. They are all very distinct individual viruses, and they are all completely unrelated. So some of them are DNA viruses, some of them are RNA viruses. Chronic infection, the two most serious are probably hepatitis B and C. And chronic infection with either of these can cause liver cirrhosis and eventually hepatocellular carcinoma or sort of liver cancer. Both of these are commonly transmitted via sort of blood-to-blood transmission. The main risk factor for hepatitis C is IV drug use, and quite a few people also contracted it from pre-1990 blood transfusions, because the virus is only discovered in the late 80s and there wasn't an effective screen for it before then. Hepatitis B it's mostly vertical transmission from mother to child. Hepatitis D is quite interesting, depending on how you look at it, because it can only replicate if it's in the presence of hepatitis B. So you can only have hepatitis D, or sometimes referred to as hepatitis delta, if you've got hepatitis B, and it makes the infection worse. And hepatitis E is different from the other two, as it's mostly spread via sort of contaminated food or water. In the UK, lots of pigs have it, and some seafood has it as well. It's quite rare in the Western world, but in sort of Asia and parts of that, it's a bigger problem. And it causes a self-limiting sort of acute mild hepatitis, but for pregnant women and for the immunocompromised, hepatitis E can be lethal. So these are just some graphics to show you sort of how hepatitis or viral hepatitis is a growing problem for the world. The first graphic here shows the number of people killed by various diseases, and as you can see, viral hepatitis has moved from the 10th in the 1990s to 7th here, which is fairly unique apart from AIDS in communicable diseases, as the only one that's on the rise compared to malaria and TB, for example. And also viral hepatitis does kill more people than AIDS, TB, or malaria. The graph on the right, interesting, shows that it's not you would think it would be a problem for the developing world and for the things, but it's actually also a problem for the Western world, and actually kills more people in the Western world and in the more developed countries than it does in less developed countries. This is probably due to the fact that it's a chronic disease, and people in less developed countries are dying of other things before they are killed by their hepatitis. But it's an interesting statistic to show that it's a problem for the whole world. As this next figure is going to show, it's just a graph showing the sort of distribution of hepatitis. So you notice I've not really talked about hepatitis A, and that's because it's not really a problem at the moment, because there's an effective vaccine for hepatitis A. Hepatitis C and B are the two main killers in the world, and sort of you do have sporadic bits of E in India and Southeast Asia. So the bit I'm here to talk about as well is the full title of it would be the National Institute for Health Research Health Informatics Collaborative, but I'm going to shorten that to NIHR HIC for the rest of the thing, or just the HIC for the rest of the presentation, as we've only got half an hour. But the NIHR is an organisation in the UK to fund medical research within the NHS, and as part of that, they funded 5 biomedical research centres, which are collaborations between NHS hospital trusts and universities. And as you can see below, there's below the NIHR logo, these are the organisations. So there's Oxford University, Cambridge University, University College London, King's College London, and Imperial College London, and their various healthcare trusts. and they form the biomedical research centres in the UK. As part of that, the Chief Medical Officer in the UK, Dave Davis, laid down a challenge to try and integrate healthcare records or parts of healthcare records to facilitate academic research in these organisations. So as we move on, the NIHR HIC objectives were to produce exemplar research projects. So they wanted to produce actual research and publications and get data out there. To do that, you need to provide the modern infrastructure to allow this data to be collected and to be integrated. There are various challenges with that, which I will go into in the future in the next slides. And by doing this, we want to establish rich longitudinal data sets of patient information over time in five disease areas. The one I'm going to be talking about today is viral hepatitis, but there are others such as acute coronary syndrome and various cancers. Within that, we wanted to develop a common governance and PPI thing with consent. So we're collecting this data mostly without consent, but this is anonymized data used to create large data sets. So it's impractical to get consent of every single patient that comes through the hospital with viral hepatitis. So the logic behind this is by using anonymized data, we can leverage the large volumes of patients' data to generate new and novel research. And I won't go through the rest of the objectives, but they're basically around sort of improving electronic healthcare records and trying to better integrate the BRCs to work better as a group. There's challenges involved in all of the themes, but there's some specific ones with the viral hepatitis theme. We were trying to collect a very large data set. We're basically talking about 4 or 5 separate diseases here with distinct research questions and distinct problems and distinct sets of data that we need to collect to answer those. So this is a diverse set of data, which is very large compared to some of the other projects which are collecting smaller amounts of data, but on larger groups of patients. There's 5 different NHS trusts, which means that all 5 trusts had different electronic patient record systems. They store the data differently, they collect it differently, but also it's the operational differences as well. The trusts all run different clinics. They offer different services to their patients because their patient populations are different as well. For example, Imperial College mostly focus on HIV patients and IV drug users. UCL have a big pediatric focus and have a lot of people or women with hepatitis B who are pregnant. So the data that they collect and the patient settings are all going to be quite unique. So integrating that was quite a big challenge, and trying to just rationalize what data we're going to get from where was quite a large challenge. Research in hepatitis C specifically, and also hepatitis B more so now than a couple of years ago, is extremely fast moving and very competitive. So we had to carve out a niche with this project. We couldn't just do what everybody else was doing because they would be able to do it much quicker than we could. So the unique factor that we have is that we're collecting a large longitudinal data set with theoretically a denominator of all the patients that are in there. So in these 5 trusts who have hepatitis, giving us ability to look at how the disease is transmitted, how the distributions of demographics of patients within these trusts that other studies within the UK and the world can't necessarily do. There is also a unique-ish problem with viral hepatitis, that these are vulnerable groups of patients. There is still a stigma attached to some types of hepatitis, and a proportion of these patients will be co-infected with HIV as well, which has a similar stigma. So there's a need to protect the patient's information and the patient's interests, and we've effectively done that by we've gone to research ethics approval in the UK and have been granted ethical approval, but we've had to go through various safeguards to make sure that the data is used in appropriate studies and appropriate work. The data is all anonymous, which helps with the ethical approval. Research questions that we decided to try and answer initially, and we're in the process of answering now, are with Hep C. These are quite broad questions initially, but we hope to focus them down once we have a better understanding of the data we've got. Is in HIV and Hep C, what influences the treatment outcomes with new direct antiviral drugs? And in hepatitis B, what are the determinants of hepatitis B S antigen and E antigen loss and seroconversion? They're markers for sort of treatment milestones. If these things happen, that's a good thing in your Hep B treatment. And in hepatitis E in the UK, it's as simple as who gets hepatitis E and why, because we just don't know a lot about what happens in the UK. So within this, there's when the project was first set up, Professor Jim Davies, who sat in the front row, came up with this sort of general conceptual architecture for how this project would work. And I'm sort of going from the bottom down to sort of explain. The bottom sort of row of this is the operational systems of the trust. So you've got your electronic patient records, your laboratory management systems, your pathology systems, your imaging systems. They will feed the appropriate data into a theme-specific database, which is the database that I'm going to be talking about today. And this will feed into a database collecting all the other data from the other themes. And there will be a metadata registry of this data to provide to researchers to say, this is the data that we have and how you can combine that for your research project. And then once that's done within the individual trusts, that will then be combined in similar databases and metadata catalogs for the whole project of integrating. But today, I'm going to this was just to give you an overview of the behind the project. I'm mostly going to be talking about this section here. To follow on, this is a slightly bigger sort of a focused-in view of that area. And at the bottom, we have again that sort of operational systems, where you've got your sort of diagnostic, your therapeutic systems, and your sort of LIMS systems, and they will be feeding information, passing through messages between each other through the trust integration engine, which we use a system called MRF Connect to do that. That data is then skimmed off into a clinical data repository, which collects this data in a longitudinal record for each patient for the trust, and we can then use that data for research that's collected in a standardized format that we can then pass out to other systems, and in this case pass out to our hepatitis clinical research system to facilitate research. We also had have a patient portal where patients can fill out small surveys on iPads or on computers and stuff, which the data from that is also provided in there and then loaded into our system. We also have sort of patient notes that are things that are going to be manually entered because as much as everybody wants the NHS to move to a paperless system, that's not quite happened yet and probably won't happen for entirely for another couple of years. And we want to export reports and stuff out of here. And then this data again can be passed off to various other systems for specific research requirements. So that was the general gist of what we wanted to do. And we were mostly talking about how we developed this database, which is what we used LabKey for. The requirements for that database are initially quite simple. So we just wanted to import legacy data. And it's mostly, I think most people have had this problem previously before, is mostly Excel spreadsheets that are sat on people's computers forever, and we want to sort of integrate and combine those into a centralized repository. But we also had some legacy systems that weren't used very much, but still contained some important data, so we wanted to export and import that. We wanted to be able to easily update the data sets to reflect the changing research questions. So while the NHIC data sets were set, fairly rigidly set, we wanted to be able to, in this system, update and add in additional fields and additional data sets to answer new questions that came up or researchers had. We needed robust security and user roles, as we're going to be collecting patient data within the system, so we need to keep that safe. And we wanted sort of customized reporting and exporting of data. And LabKey took care of these. The standard build of LabKey takes care of all these things pretty well. So we were fairly happy with that, which is the reason that we chose the product. So the next two things is something we had to do some sort of custom development for on our own side, is a patient-centric data manual entry and editing of clinical data, and I'll explain a little bit more what I mean by that in the next couple of slides and how we address that problem. And automatic importing of data from our middleware into our LabKey system, which we were actually using fairly standard LabKey ETL processes we were able to achieve. So patient-centric data entry. The standard model or workflow for entering data manually into a LabKey dataset is quite dataset driven. So you open a dataset, you add in a row, you add in the next row, you add in the next row. That's not quite how nurses work or how our people who are entering the data work. They'll take a set of patient notes and they'll go, we want to add a row into this dataset, this dataset, this dataset, and this dataset. And that, in the standard model that LabKey provides, it requires a lot of clicking and back and forth. So we decided to try and develop our own module for entering data manually into a LabKey dataset, which allowed people to follow this more patient-centric model rather than a sort of data-centric model. So I've just here to just demonstrate what the standard LabKey interface for entering data looks like. So we developed, or one of our editors developed a module which he decided to name LabKing with the tagline "Rule over your subjects," as it's a subject, a sort of participant-specific thing. So it was built using the LabKey JavaScript API, using sort of an Angular application, and can be used with any data sets and uses as much as possible the standard features that LabKey provides. So we were trying to do the minimum amount of hard coding of specific fields that we wanted to enter, and we've largely achieved that, where if you change a data set in the standard LabKey flow, that's reflected in the view that you get here. And this is done, and for anybody, you can have a look on GitHub. The code's there to download if anybody's interested. And we'll be happy to try and explain a bit more to you if you wanted to. So the next thing I've got is a video which hopefully will work, sort of showing what we did with this. I think it's not that clear on the screen, so I might not linger on here too long. But first we have a dynamic search feature. So as you type, you can search for any of these are field names in an enrollment data set we have, which contains all the identifiable data from the patient. So we have patient name and then various identifying numbers that we use within the trust for that, as well as date of birth. And this search function just searches for all of those and gives a sort of dynamic search for that. So when nurses are trying to pick the patient they're interested in, they can just find that and click on it. This is the LabKey cohort feature. So the cohorts that are in LabKey are reflected here, and you can sort patients by the cohorts they're in. So you can just tick the cohorts you're in, or select all, or select things. We also do that with participant groups as well, but in this build we didn't have any participant groups. Once you've done that, you can then just select a patient, or actually you can add new patients. I'm not sure if that's hidden in the corner there, but there's a button there to click Add New Patients, and you're presented with this form, which is just the LabKey dataset form. So we provide date pickers and drop-down boxes and free text fields for those to enter. So you can go through the form, fill out the information, and then just click Submit, and that's sent off straight to the database. So once you select a patient, the module goes off and loads in all that data for that patient. So over here on the top row, we have the data set categories that you set in LabKey are the top set of tabs for each individual data set. And then as you click on one of those, the data sets within that category are shown below. You can then see and view the data in this table here, and then click on an edit or a delete button to add or delete that data set. So you can just click through each data set relating to that individual participant and enter or edit the data as you see fit, and that data is then just updated in the database as you go along. Once you sort of solved that issue, and that was something that our nurses are now using on a day-to-day basis to enter patient data and to edit it, we wanted to approach importing the data from our other systems, and we used a fairly standard, fairly simple approach. Where we're using shared tables and just loading using the ETL process in and out. So the hepatitis clinical research system passes a list of participants that we're interested in to our middleware, which then collects all of the data that we're interested in on those patients, sends it to another shared table, and LabKey then loads that up, and that happens every night at midnight, so we get a daily update. There's another slightly more interesting way that we do it as well, is that the clinical data repository can search for patients they're interested in. So we want a positive hepatitis C test from the hospital laboratory. It will find all of the positive hepatitis C tests and those related patients and pass that list of we refer to them subjects of interest, to the hepatitis clinical research system. Then nurses can go through and select the patients that they're actually interested in. We may eliminate that manual quality control check at some point in the future, but at the moment we just want to make sure that everything's working correctly. And then once they're into the system properly, it just goes through the same cycle to load the data in. So it's a fairly simple process, and it was actually quite simple to set up using the LabKey ETL process. So showing you the previous diagram I've just shown you with a little bit more sort of information filled in about where we're, of how we did stuff, is there are about 30,000 DPR messages sent every day between the various operational network things. There's about 3,000 microbiology messages and 20,000 LIMS messages. All these messages are read and passed into the CDR, loading the appropriate data into longitudinal records for those patients. So this is a fairly old number now, so I'm not sure I'm sure it's probably higher than this now, but there were 60,000 unique patients. Also 600,000 unique patients in that system, and then the patients that we're interested in using the cycle I've just shown you are then loaded into the LabKey system. The advantage of the system we did, of the automated system that we showed you before, is that because we're using shared tables, we can drop in another system on there as well. If this system gets replaced at any point, then we can just drop in another system and it will continue to work the same. So far we have about 1,200 patients loaded into there. That number grows by about 100 every month or so, or depending on the month. And we get about 30% of our data from manual entry and about 70% of it, mostly lab results, loaded in automatically. And we've had 78,000 lab results loaded in so far. So this is a huge data set in the hepatitis world for lab data. Previously, if a study wanted this amount of data, like lab data or lab data for a hepatitis research study, the research nurses would have to go through manually and pick out each one and type it into an Excel spreadsheet. So there is no way that you would get research nurses to do 78,000 lab results manually. So this is a really unique resource that we have here to be able to do research. And then that information is passed off to various other systems. So we pass it off to the HCV Research UK database, which is a separate hepatitis research database, which was a system that our nurses were entering data into manually, but now we can provide it in a slightly more automated fashion. We pass it off to the NIHR HIC Hepatitis Research Repository, which is the project I was talking about before, where it's integrated with the other trusts. And we also pass it down to the Oxford University Pathogen Research Lab, which is the lab that I work in, where we're building another LabKey system to try and integrate the research lab data that we're doing down there, with viral sequencing data, with immunological data, into this, with the clinical data that's there. And that's a really unique resource as well for some of the researchers that are working down there, because they previously would have to go to the research nurses and go, can you get this data on this patient? And they would say, yes, we can, but it's going to take 2 or 3 weeks because I've not got the time to go and do it now. Whereas now they can log onto this system. We provide them an anonymized review of this patient's data with the IDs that they're given for research, and they can just load it themselves, and they can export it, and they can do their research. So it's speeding up processes within the research environment. And the more people use it, the more they're interested in it, and the more requests I get for people to set them up new data sets or new LabKey installations and stuff. So it's working, and it's slowly building a bit of momentum. So these are some of the ongoing research projects that we're looking at. Just the small projects. This isn't the big NIHR HIC projects. These are the small ones we're looking at just in Oxford. And it's things like instances of hepatitis E with underlying liver disease in the Oxford cohort. So the hepatitis E is a fairly rare thing. It's even more rare in Oxford, and the doctors just weren't, unless they saw the patient themselves, they just didn't know how many people were coming through with hepatitis E, because there wasn't an easy way to look without somebody sitting down and going through all the records. Whereas now we can just run off a report every month and say, look, we've got this amount of positive hepatitis C results this month, and we can look at the connections between other liver diseases, or things like where did they travel, or what pork products did they eat, or stuff like that. So this is like a really important thing for a really small scale study, but something that just wouldn't have happened otherwise. We're integrating longitudinal clinical data with viral and host genetic data. This is a really sort of cool stuff that we're doing as well, down at the Medwall, where we're integrating both host genetics and viral genetics. But that data is not that useful unless you have the clinical data to go with it. If you can't group patients by their liver disease status and that sort of thing. And again, that data was a lot more difficult to collect before this system came about. There is work going on to develop a hepatitis C vaccine. In fact, there are clinical trials going on now at the moment, but this is they're not using this system as much as the other projects, but it is something that we're looking at trying to expand a little bit more and try and develop a bit more connections with that team. And then this is the work that I'm doing on my PhD, is integrating the clinical data with viral sequencing to better understand drug resistance in hepatitis C. And you couldn't do this without the clinical data that's there. You can have the viral sequencing and you can go and know that this virus is resistant, but what does that mean unless you can apply it to clinical data about what drugs they had, when they had them, what stage of liver disease that they have. So this is quite a nice follow-on for me from helping to develop the system to now using the data in my own research. Some of the future work we're working on is collection of the minimum data set for the NIH-HICS. So that's all the individual sites providing their data so we can analyze it. And we have quite a large volume of that data already. We just want to try and make sure that we've got the highest quality of data, rather than just having a huge volume of it. We also want a huge volume of high quality data. So that's something we're working on. Obviously the analysis of that data for the research questions, and then we're working in some partnerships with industry partners on doing collaborative learning. So this is a fairly new thing that's just started happening, but there are a large amount of drug companies and other companies in the UK who are interested in this data set, and we are interested in working with them in a collaborative research environment. And then the further developments of the system, we want to do some more reporting and stuff, so we do a limited amount of reporting, but we'd like to expand that. And we'd also like to develop a sort of dashboard view for clinicians, so what clinicians, not necessarily the nurses, but the doctors who are doing research want to see is just want to log on and quickly look at a thing of how many Hep C patients do we have in there, how many of them have had this drug and that sort of thing. And we want to sort of develop a dashboard that can just show them this key information as soon as they log on. We want to further integrate that with other clinical systems that are being used. So we want to do some more imaging stuff and some more pathology reporting. As I was talking about something earlier today, that the pathology reports come in a free text format, which makes it quite hard to pass the data into the system. So we're going to look into sort of developing that slightly more, and also the imaging data and whether or not we can load that in, or what we actually want to load in as well from the imaging stuff is something we're looking to explore. And then automating the quality control is something we're looking at. So once this system has reached project has reached its end, we want to look at trying to automate as much of the quality control as possible. So when there aren't people like me and other people who help design the system around, that it will continue to generate high quality data. So that's me coming to the end. There's a long list of people in here, but I've missed off more people than I've put on. So there's mostly people within our group. Jim and Charlie are sat in the front row, but there's lots of other people here. So specifically Ryan Brooks, he was the developer who developed our LabKing module, and his Sahil, and another guy that I haven't put on there called Mark, who developed the CDR stuff and the interface between these two systems. And the system wouldn't exist in its current format if it wasn't for those two guys. These are the clinical leads from the NHICS project who have organised various things, and there are this amount of people, again, in all of these departments who are working on this sort of thing. The hepatology research nurses who have been invaluable in their feedback of, we don't like this, we do like this, can you change this, can you make this button blue? That's essential and will be essential in our further development. And I've only mentioned 3 people on the LabKey team, but lots of other people have helped in those things. But these are the people I've most been in contact with, especially Bernie, who continues to come and speak to me on Skype every month. Yeah, thank you. And also all these organisations who are involved in the project.